to you.
Thank you. Good afternoon, everybody, and thanks for being here today. In the next 20 minutes or so, I'm going to explain to you why I find fossilised faeces so fascinating, what they can tell us about the way animals lived in the ancient past, and even how they can help us better understand past and current climate change. So let's start off with some pictures of coprolites. So you can see that they're quite varied. These are coprolites from all sorts of different animals, and some of them, like these two, really look exactly how you might picture fossil poo to look. And then others, like these ones, they really just look like lumps of rock. And the way that you can tell if a suspicious-looking rock is actually a coprolite or not is if it contains fragments of partially digested food, which might be either plant or animal material, depending on whether the animal that produced the coprolite was a herbivore or a carnivore. So in the fossil record, most of the coprolites that we find are from carnivores, like this one here, which is from T-Rex. And this is actually the biggest coprolite that's been found so far. This is over 30 centimetres long, and it weighs about 7 kilos. And the reason that, that most coprolites that are preserved are from carnivores is because the minerals in the bones of prey animals, which end up in the faeces of carnivores, the minerals from the bones kind of predispose the fecal material to lithification, which is a process of turning into rock. And herbivore coprolites don't have all these minerals in there, so they need a much more kind of special set of environments in order to get preserved. So carnivore coprolites are big, there's lots of them, and they are pretty much the most spectacular coprolites that we find. But all of my research is based on herbivore coprolites, which have got much more interesting chemistry. So the word coprolite, which literally means dung-stone, was coined in 1829 by William Buckland, who, by the way, was a fellow of the Royal Society. And he invented the word coprolite to describe some rocks that he looked at, which were like this one here, rocks that had been collected from the beach and the cliffs in Lyme Regis in Dorset by Mary Anning, who was one of the very first female paleontologists, and who was quite unusual in working as a paleontologist at a time where women really didn't have the opportunity to get very involved in science. So Mary Anning collected a lot of these types of rocks, which she called bezoar stones, and they were mainly collected close to or associated with the skeletons of marine reptiles. And when William Buckland looked at the bezoar stones, he found that they contained fossilised fish scales and fish bones, and he realised that they were actually fossilised digested material. So since then, since the time of William Buckland and Mary Anning, scientists have continued to study coprolites. But why do you think we bother? What do you think could be the reason for being interested in coprolites? Any ideas? Yeah. They might do. I'm not sure anybody's said that yet, but it could do. Any more ideas?
Tell how big the dinosaur was, whether it was carnivore or herbivore.
Yeah, that's right. Any more?
Tell you what they ate.
Yeah, that's the main one. Anybody got a different answer?
The chemicals that were mainly dominant at that time.
Yeah. So there's all sorts of interesting information in the coprolites, but the main one is the diet. And the other thing about studying coprolites and other trace fossils, which are fossils that are not the actual body parts of an extinct animal, is that they're one of the only direct pieces of evidence that we have for actual behaviour or kind of activity between animals in the geological past. So, for example, if you find the bones of animal X in the faeces of animal Y, then you can tell that there was definitely a predator-prey relationship between those two animals. And that kind of direct evidence of behaviour is actually pretty rare in the fossil record. Yep.
How do you know which of these faeces come from which dinosaur animal?
Oh, that's one of the that's a really good question, and it is one of the big problems in working with coprolites in general. It's actually well, you'll see in a couple of slides. Sometimes we've got really direct evidence for it. Sometimes we haven't got any idea. Some of the coprolites on the first page haven't been attributed to a producer. And sometimes, like with the T-Rex coprolite, we've got kind of circumstantial evidence. So with the T-Rex coprolite, who was it said about the size of the animal? Basically, the T-Rex coprolite that I showed the picture of, that was found in Canada. And in the general area, the only large carnivorous dinosaur remains that have been found are T-Rex. And so we make an inference that large coprolite has come from that large animal. So it really depends on the specifics of the coprolite itself and the environment that it's been found in. So most scientists who've worked on coprolites have looked at the size and the shape and the food particle content. But the research that I do is slightly different because I look at the fossilised molecules that are preserved within the coprolites, which are called biomarkers. So biomarker is a term that gets used quite a lot across a lot of different areas of science, and sometimes it's used in slightly different ways. But in organic geochemistry, which is a field that I work in, a biomarker is defined as a molecule that can be attributed to a biological source on the basis of its chemical structure. And so this is based on kind of modern occurrence. So for example, if you find a molecule in a plant or an animal or a microbe today, and then you find the same molecule in a rock, we assumed that the molecule was produced by a similar organism. And this is called the principle of uniformitarianism. And this is one of the fundamental principles of earth science and paleontology in particular. And it's sometimes summed up by the phrase, the present is a key to the past. Ooh, sorry, I should just say as well, most biomarkers, but not all of them, are molecules that are associated with the cell membrane. So, for example, we've got fatty acid chains here, which are part of the phospholipid bilayer, and cholesterol here, which is a molecule that helps to kind of strengthen the cell membrane. And these molecules, and other very similar ones in plants and microbes, are some of the most common types of biomarkers that we find. So biomarkers and faeces come from three main sources. And the first and the most obvious one is the diet of the animal. So in the case of herbivores, these are molecules that come from the plants that the animal eats. Secondly, we've got the molecules that come from the animal itself. So for example, cholesterol, which is found in animal cells, often makes its way into herbivore feces as the food passes through the digestive tract, and some parts of the cells that line the digestive tract are kind of scraped off and incorporated into the feces. Also, there are molecules that are directly involved in digestion that the animal produces. So, for example, bile acids, which are essential to digest fats. And then finally, and for me the most exciting group, are the molecules that come from the digestive tract microbes that live in the gut of all animals. But in herbivores, in particular, you get a really diverse range of microbes. Okay, so I've just got a little bit about how we actually analyse biomarkers from coprolites. And I've just got I've got a coprolite and a powdered coprolite here. If you want to have a closer look, if you just want to pass that around. So we start off with a dried sample. In the case of coprolites, the samples are already dry. But when I work on modern feces, when you get them kind of fresh from the farm or wherever. Have to dry them out first before we can work with them. And then the next thing that we do is we basically destroy the sample by grinding it up into a powder, which we do the old-fashioned way with a mortar and pestle. And so the two samples that are going around now, we've got an intact coprolite, and then we've got the powdered version as well. The next stage is to extract the organic molecules from the powdered coprolite. And we do this using organic solvents on the principle that like dissolves like. So organic molecules will dissolve in organic solvents. And this is basically like making a cup of tea. So when you take your tea bag and dunk it in the hot water, then all of the water-soluble compounds that are in the dried powdered tea will go into solution, will dissolve in the water. And it's exactly the same thing with the organic solvents and the powdered coprolite material. So all of the organic molecules dissolve in the organic solvent. So then we end up with a mixture of molecules, which we separate into groups according to their chemical properties. So, for example, the functional groups that they have. So, for example, we can isolate all of the alcohols from the mixture. And we do this using column chromatography. And then finally, we analyse the samples by gas chromatography and gas chromatography mass spectrometry. And this allows us to quantify and identify the molecules from the coprolites. So this is one of the first coprolites that I analysed, and it's a sloth coprolite. It's about 11,000 years old, which is quite old, but not by geological terms. And this coprolite comes from this species of ground sloth, which is called Nothrotheriops shastensis. And I don't know, has anybody seen the film Ice Age and all the sequels? Yeah? Remember Sid, the giant ground sloth? Okay, well, this is only a medium-sized ground sloth. This isn't a giant ground sloth. This one was probably about the same size as me. And going back to your question from earlier, in this instance, we know very specifically that this coprolite came from this species. This material was found in a locality called Gypsum Cave in Nevada in the US. And this coprolite and lots of similar ones were found next to the body remains of this animal. So that's already a good hint that that coprolite was produced by that animal. But what really clinched it is that this coprolite was so well preserved that we could even extract, well, not me, but other scientists could extract DNA from both the coprolite and the physical remains of the animal and match up the DNA to demonstrate that this fecal material was produced by this animal. But that's quite an unusual situation. Normally it's much more difficult to work out who made which coprolite. So we knew from various bits of evidence, including the DNA studies, that this particular animal was a herbivore. And so we expected to find plant biomarkers in the coprolite. We expected particularly to find phytosterols, which are the plant equivalents of cholesterol. So they do the same job in the cell membrane that cholesterol in the cell membrane of plant cells that cholesterol does in animal cells. And we did find some phytosterols, but we also found an awful lot of this chemical, which is called Epismilogenin. And this puzzled us for quite a while. It took us quite a while to work out what it actually was, and then to find out where it came from. And Epismilogenin is a spirochetal sapogenin. If anybody is into the chemical structures, this is the spirochetal group here. And Epismilogenin is formed when plant secondary metabolites are digested. So secondary metabolites are compounds that plants produce that are not part of their essential functions. So they're kind of extra molecules that the plants make. And And for example, sometimes plants make secondary metabolites in their leaves that have got a bitter taste as a way to try and protect themselves against being eaten by animals. So we looked at published reports, and it turned out that there are only 2 types of secondary metabolites that, when they're digested, are converted to epismilogenin. And then when we looked a bit further, there are only 20 plants that make either of those 2 compounds. So we were kind of narrowing down the source for the epismilogenin. But then when we looked at the distribution of these 20 plants across the world, there were only 2 plants that were in the right place at the right time that could have been the source of the epismilogenin in the sloth dung. And they were yucca and agave. And so we conclude that the epismilogenin found in the sloth coprolite was produced by the animal eating a diet that was really high in either yucca or agave, or possibly both. Now, if you look at them here, they look pretty spiky and not at all pleasant to eat, in my opinion, but obviously sloths felt differently. So, so by looking at the sloth coprolite, we were kind of reassured that, in fact, biomarkers were preserved in coprolites and that they could provide some interesting information about the diet of extinct animals. And so we decided to go back to modern animals and look at fecal biomarkers from a whole range of modern herbivores in order to find out what factors controlled the variety of biomarkers in herbivore feces. And so although herbivores only eat plants, they don't actually have the right enzymes, or they can't actually make the right enzymes to directly digest the plant material themselves. And they're obliged to live in symbiosis with microbes who can digest, particularly cellulose, which makes up the large part of plant cell walls. So herbivores all have a large, quite complex microbial community. And this includes fermentative microbes that ferment the plant material and produce volatile fatty acids, which are a food source for the animal, and they also produce hydrogen as a byproduct. And herbivores generally fall into one of 2 categories according to the structure of their digestive system. So foregut fermenters, like cows, sheep, camel, giraffes, also deer, foregut fermenters have got a special chamber to house their fermentative microbes that's situated near the start of their digestive tract, just here. So you can imagine the mouth is over here somewhere. And this is the fermentation chamber here. And then this little structure just underneath is the animal's true stomach. And hindgut fermenters have got the opposite arrangement. So if the mouth is over here, this is the true stomach that produces enzymes. And then further down the digestive tract, you've got the fermentation chamber, which is called the cecum. And this is where the microbes are situated. And this has got implications for the way that food is digested. So basically, for foregut fermenters, the microbes get the food first, and the animal's own enzymes digest what's left over. And then in hindgut fermenters, it's the other way around, and the animal's own digestive enzymes get to work on the food first, and then the microbes ferment the residue. And because of this quite fundamental difference, we thought that we might see a difference in the fecal biomarkers between these 2 groups of animals. So we analysed feces from all of these animals and quite a lot more other species as well. And what we found was that this molecule, Archaeol, was found in the feces of foregut fermenters, but not hindgut fermenters. And Archaeol is a biomarker for a group of microbes called Archaea. And there are lots of different types of Archaea that all perform different functions. But in herbivore digestive tracts, the only types of Archaea that have been found there are ones that produce methane, which are called methanogens. And so when we find archaeol in faeces, we interpret that as a biomarker for digestive tract methanogens. And the way that the methanogens fit into the biochemistry of digestion in herbivores is when the fermentative microbes produce the volatile fatty acids and the hydrogen, the methanogens use the hydrogen together with carbon dioxide to produce the methane. So once we found archaeol in the faeces of modern foregut fermenters, the next thing we wanted to do was to see whether archaeol could be preserved in coprolites. And this is the oldest material that we've worked on so far. It's not geological age. It's only 2,700 years old, and it comes from the Yanghai tomb in China. And it's a grave, basically, a tomb. And this is a guy who's been buried along with a lot of pots and other equipment. And he's also buried together with some animals. We know that the animals were alive when they were buried. We've got the animal bones. But we know that they were alive because there's actually quite a lot of coprolites in the tomb as well. So the animals were alive for some time, producing feces before they died in the tomb. Where did the samples get to? Has everybody seen them? Great. Okay. So what the you'll notice on the labels there that they're labelled as ovicaprid feces. This means that we don't know whether they came from sheep or goats because we've got bones from both of those animals in the tomb. If we wanted to, if it was important to the archaeologists, we could use DNA analysis to distinguish, but that's not something that the archaeologists who investigated this tomb were interested in. So this is the oldest material that we looked for archaeol in, and this is a result of our GCMS analysis. So each of these spikes here, each spike represents a different compound, and this little tiny spike over here is archaeol. And this is the mass spectrum of archaeol, which allows us to identify it. So now we were thinking that, okay, we've got a biomarker for foregut fermentation. We can find it in ancient material. But then we stopped to think, and then we decided that actually it wasn't as straightforward as that, because hindgut fermenters also produce methane. They produce a lot less than foregut fermenters, but they do produce it, and so they must have methanogens in their gut. So why didn't we see any archaeol in the hindgut fermenter feces? And this made us start to wonder whether there was a quantitative relationship between the amount of methane that an animal produces and the amount of archaeol that you find in its feces. And we decided to test that on modern animals. So we, together with some with colleagues, we carried out a feeding experiment on some cows, and we had two groups of cows. One group had a silage diet, so lots of grass in the diet, and they produced quite a lot of methane. This is the cows on the grass diet. And the other group had a diet that had a lot more concentrate feed in it, and they produced much less methane. And then we analysed the amount of archaeol in the faeces of these two groups of cows. And what we found was that the animals that produced a lot of methane also had higher levels of archaeol in their faeces than the animals that produced less methane, which were the ones on the concentrate diet. So it does look like there's a relationship between the amount of methane that's produced and the amount of archaeol in the faeces. But why is that important? Well, it's important because methane is a greenhouse gas and it contributes to global warming. And over 60% of global methane is derived from human activities, which includes farming. And about 20% of the total of all the methane that's in the atmosphere at the moment comes from domesticated ruminants, so farm animals like cows and sheep. And we have a reasonably good idea or an estimate for the amount of methane that's produced by these domesticated animals. But one thing that we don't know is how much methane is produced by wild ruminants. So, for example, herds of bison and buffalo, or the wild camel population in Australia. And at the moment, we haven't got any real way of measuring that, because the current methods of measuring methane emissions from animals are actually not suitable for wild animals, because, as you can see in this picture, they involve quite close contact with the animals themselves. So the earliest developed method of measuring methane emissions was to put the animal in a sealed chamber and measure all of the gas that came in and out. And a slightly more recent method was to use the gas sulfur hexafluoride as a tracer. So you persuade the animal to swallow a canister of this gas, and the gas is released at a constant rate. And then the animal wears a head collar, which is a red and white stripy thing, and this takes gas samples, and by comparing the amount of the sulfur hexafluoride to the amount of methane in the gas samples, you can work out how much methane the animal is producing. And the reason that the animal's wearing a head collar is because about 80% of the methane produced by cows is actually exits the animal via the mouth, via burping, basically. And only 20% comes out of the other end, contrary to popular belief and what you read in the newspapers. But neither of these methods are suitable for wild animals. But if we can develop this correlation between fecal archaeol and methane emissions, then that would give us a method that we could use to measure methane emissions from wild animals, because we would only need to collect their dung, so we wouldn't need to disturb the animal at all. So we're not really at that stage yet where we could go and do that, but this is what I'm going to be working on for the next couple of years. And if we do manage to develop this technique and get it working really well for modern animals, using fecal archaeol to estimate methane levels, then there's also the potential that we could expand that and apply it to the geological record. So that would mean that we could measure archaeol from ancient feces and use that to estimate how much methane an extinct animal might have produced. Okay, so just to sum up, what can coprolite chemistry tell us about extinct animals? Well, it can tell us about the diet, can tell us about their digestive processes, and it might be able to tell us about their methane emissions. And I just want to finish by stressing that this is really still a work in progress. This is a really new area of science, and every single coprolite or fecal sample that we analyse gives us more information and helps us to better understand these processes. Thanks.